Let's look at some of the physiological interactions that occur. First, it says how do pathogens establish infections? There are a bunch of other things that we've got going on in here at the same time. Let's look at some of the stuff that happens. We're going to deal with enzymes. This is an apple. It's been attacked through the flower end. This material has caused a liquefaction, a modification of the tissue. This is all due to enzymes that occur by the pathogen that are degrading the tissue so that the pathogen can get nutrients. This shows you a peach. You can see the enzymatic degradation of the peach starting down at the infection end, down at the bottom, and migrating upwards. This looks like a papaya. You can see the enzymatic degradation of the papaya area by the pathogen. This is an apple. You can see the enzymatic degradation of the apple beginning at the infection end and moving inward. This will liquefy the entire fruit. I remember hearing a story about a car of potatoes that was shipped out of Idaho that had a problem in the refrigeration system. It had been on the tracks for a week getting to Chicago. By the time it got into Chicago and they opened up the railroad cart, the entire thing had liquefied. Enzymatic degradation is very important in being able to help the pathogen get in. Enzymes can go out and degrade tissues on the inside. They can affect the internal structure of the tissues and cause for things like wilting. There are toxins. When we deal with toxins, we call them phytotoxins. We've got two types. We've got a general type. General type's going to work on anything. The specific type's going to work on a very specific pathogen. In this particular case, we've got corn. You can see the symptomology on the corn. This is due to a toxin. This is a toxin on Digitaria. This is crabgrass. Crabgrass is normally a very important weedy species. You can see around the outside how it's got these purple halos, how the halos actually extend up. That is toxin being produced by the pathogen. You can look at the stem end in the upper left-hand corner. You've got the toxin migrating out towards the end of the plant. You can see how it has affected the symptomology. All of this becomes very important. We have got growth regulators. When you're dealing with growth regulators, we've got things like gibberellic acid and we've got auxins. You've got the release of apical dominance. On the left-hand side, you've got a carrot. I know it doesn't look like a carrot. What happens with carrots is if you disturb the primary root meristem, you will get release of apical dominance and you get all of these secondary meristems coming off of the sides. You get this. This could occur from nematode damage. This could occur from transplanting. I had a small area in Brazil where we were growing carrots. The person who did this, rather than planting the seeds, went out and put them into little nurseries and transplanted the carrots. In the end, I ended up with 500 kilograms of carrots that looked like this. Absolutely worthless on the market due to a growth regulator. You can see growth regulator on this. Looks like a cucumber type plant. You're getting modification of the meristem due to the modification of the hormones or the growth regulators that are on the inside. When we look at plants and what goes on with them, there are a lot of different chemicals that can be important in the development of symptomology or pathogenesis.